Thank you, Thibaut, and thank you for the chance to giving this talk this morning in this school. So hopefully at the end of this talk you will understand, well, I will try to convince you that all the theories that Denis has provided you are probably applicable or, let's say, useful for understanding biomineralization of real biominerals. And so I will show you how we can use nacreous organic extract to drive this biomineralization. So it maybe it's the wrong arrow, but no, I get the right one. Okay, so as Thibaut showed you this morning, biomineralization is of course a way to bring crystalline shapes which are not found in synthetic conditions. And that could be, the reason is many functions which are provided by these shapes, which could be the protection of the animal, like in this phytoplankton. As mentioned this morning, there are also optical functionalities which are brought by small calcium carbonate lenses, and also floatability for this animal to just go down and up and down in the water thanks to that. And so we would like to understand much more about how is this formed. And the example I've chosen this morning is a famous example of nacre, and I will drive you to the structure of this, of one nacre. Actually, there are many others, but this one is the one we have studied. So when you look at this pearl oyster, and if you cut it in the middle, and if you look at the structure, what you will observe is a kind of a stack of lamellar layers of calcium carbonate. And actually, the polymorph is aragonite, with the C-axis of aragonite, which is organized in this way. So these are really oriented aragonite tablets, which are stacked this way. And of course, organics is present at a small proportion. It's only 5% of organics altogether. But if you are able to remove the calcium carbonate part, of course, a part of this organics reveals. And you can show it here on this beautiful picture, where you see that you have walls of organics which are sticking together all these calcium carbonate bricks. And what is very much impressive in this mineral is that each tablet of this aragonite stuff is diffracting as a monocrystal, which is quite surprising. And the dimensions are here. Typically, each lamella, each calcium carbonate lamella, is about something like 400 nanometers, while the organic thickness of each lamella is about 40 nanometers here. Of course, it's only one nacre, and if you look at other animals, it could be very different. This was true for this bivalve, but if you now go to different animals like gastropods, like the nautilus you showed before, you would prefer to see such an organization. It's always aragonite, but now you see it just builds up as columns and not as a brick and wall stuff like in Pinctada. But all of them are very different from the kind of aragonite you could grow in the lab in synthetic conditions. So the shape of the crystal is really dictated by the organics which is inside. Okay, but it's not as simple, because in the Pinctada you don't have only aragonite. As time goes by, you first grow not aragonite but calcite. So for the juvenile animal, the first crystal which is grown is calcite under the form of this prism. And as time goes by, at some point, the animal decides that it will change. So the shape of and the polymorph of the calcium carbonate, and it will shift from calcite to aragonite. So of course there is a whole kinetic story of the growth of this calcareous layer in the animal, which is quite complex. So the organic matter, to summarize, is able to control many things. It's able to control the location of the mineral nucleation. So for example, in these tablets. It's able to control the orientation of the crystal to the point where it could diffract like a monocrystal and limit the thickness of the mineral. And it also controls the kind of nucleated polymorphs through the organic matter. Let's describe a little bit the organic self-assembly, which is inside. We can separate in two parts, roughly speaking. where a part of this organic matter is insoluble in water, typically acidic water, and another part is really soluble in water. Generally, what one finds in these animals are an insoluble core which is made of beta-chitin crystallites, which is depicted here, which makes a big part of this insoluble scaffold that you see before when I removed all the calcareous part of the animal. On top of this chitin, you could find many glycoproteins, which are similar to some proteins you find in silk, with some dominant beta sheet conformation of these proteins. So this is for the mostly insoluble part. But there is also a large soluble part, which is included also in the tablets. So both included in the tablet and also adsorbed on the insoluble part of the organics, which is made of soluble acidic molecules, so mostly with glutamic and aspartic groups. We'll show the detail afterwards. And these molecules are either adsorbed or included in the calcium carbonate tablets. And we'll see that more precisely, these molecules, in the case of pink data, are glycoproteins or proteoglycans. So the goal, if we want to describe roughly this this organic scaffold, this was done by Weiner and Levi-Kalisman in 2001. They give us such a description, which is a core of insoluble beta-chitin stuff, which actually in Pinctada is not so much found, but in many other animals you can have it. And you have also these other insoluble proteins which are included here. And in between, you get something which is a soluble organic matrix, which resembles to a gel with some structure close to silk fibroids. So the way organics influences the growth has been studied in different ways. And one way is, of course, trying to explain the kind of polymorphs you grow. So why do you grow aragonite, which is not the most stable polymorph compared to calcite? And of course, people have tried to use the organics in order to influence the growth of calcium carbonate, starting with this paper where they claim that insoluble organics can control aragonite formation. Then it was detailed by other people till the point where groups try to isolate single molecule, single protein, and try to detect, to elucidate what is the precise role of a single protein they could isolate from the organics. And in some cases, they were able to grow aragonite thanks to a single molecules. But another question is also to reveal how these molecules are controlling the nanostructure of the biominerals. Because actually, these tablets are not so simple as you may think. And they are also nanostructured. And this has been evidenced in a quite number of papers. And I will now show you what is this kind of nanostructure. Again, when you look, and I took these beautiful pictures from Jean-Pierre Cuivre from Orsay, if you look at the nacre, you have these calcite prisms, which are first grown. Then at some point, as I'll tell you, the animal will shift to aragonite. But if you look more closely in each of these domains here, what you will find are nano domains that you could reveal by atomic force microscopy, for instance, which is here in amplitude or in phase. But the phase tells you that the chemical nature, the materials is very different within the grains here or in between. And this structure could be found either in the calcite or in aragonite. Actually, the tablets of nacre are nanostructured. It's not a single crystal. It diffracts like a single crystal, but it's made of nanograins, which are nicely packed together. And this part comes also from organics. And we would like to know why and how. So we decided to carry on study where, taking the soluble part of the organics, we'll try to understand how this soluble part is able to reproduce this. So today's question are the following. There is a kind of generic nanostructure which could be found in NAC tablets and also in calcitic prisms. And we have seen that these nano domains are surrounded by a cortex, which is of different nature. So if the center of the grain is calcium carbonate, you could think that outside of the grain, you have either organic matter, but it could be also glassy material. So the question we raise is, what is the role of this organic matrix and precisely of the intratablet? I told you it's mostly soluble molecules about that. Can it drive the orientation of nanodomains? Is there a glassy origin? So what we have done is to first extract some part of the soluble organic matrix of nacre. So we have taken this pink theta stuff and decalcify with acetic acid, centrifuge, desalting it, and then recover the part of the organics which is above 3 kilodalton. Then we can analyze this soluble matrix. We can do it in different ways. First, by destroying a part, we can measure the amino acid contents. In this process, we destroy the tryptophan, for instance, and we damage threonine and serine. But still, what we recover is a large dominance of aspartic acid and glutamic acid, which is normal. It was expected because, of course, these groups give the strong interaction of the organics with the calcium ions and drive mineralization. But also, which was also expected, glycine is very much present. In terms of distribution of isoelectric point and molecular weight, two things could be done. First, chromatography. And this is interesting because now here we have compared two animals, which is Nautilus and pink theta. And actually, you could see that for many nacre, the contribution of this soluble organic matrix in terms of molecular weight is not very different. But more precisely for pink theta, when you look at the kind of molecules which are present in this soluble extract, what you find are mostly, as I said, glycoproteins and proteoglycans, and they are mostly acidic, and the distribution of molecular weight is quite large. So there are a whole bunch of molecules of the kind of glycoprotein which are present in this soluble matrix. Then you can try to mineralize calcium carbonate in presence of this soluble organic extract. Of course, if you make a controlled experiment, so take a solution with hydrogenocarbonate and calcium salt mixed together, keep the pH around 8, and it's not a kinetic experiment. We just wait after mixing for a day or longer and get the final crystals which are made. Of course, if you don't add any organic matrix, what you recover is the typical shape of calcite crystals. So this is normal. But then, of course, there is a strong influence of the sum addition, and I will try to explain briefly what we obtain when we add more and more sum in the system. And of course, again, we just get at the end the crystals and observe them. So first we can observe them by electron microscopy, but we can also make some X-ray analysis to get the polymorph, or infrared in order to detect, for example, amorphous phase, if any. So if you increase the sum concentration, you start from exclusively rhombohedral calcite, as I showed you, but then very fast is not a good term because it's not a kinetic study, but let's say when you increase a small amount of sum, you modify this calcite and you see a new phase, new polymorph, vaterite, which is of course even less stable than aragonite, which is now present in the form of this flower-like system. So vaterite appears for some concentration, and if you now go to even higher concentration, and we went up to 100 ppm sum, and then you see that vaterite is now disappearing, and you go back to calcite, but the calcite which is quite modified, and now has not the form of these rhomboidal crystals, but a very different form, like these polycrystals that like you see here. This is confirmed by X-ray crystallography, where you see that you start by growing calcite, then get a mixture of calcite and vaterite, and then at the end, you end up with calcite exclusively. But no aragonite. Aragonite, I recall you, this is the kind of crystals that usually you get in these tablets of nacre, where we started from it originally, but we don't recover it here. Same thing with infrared, where you could first detect calcite, then of course mixture of calcite and vaterite, and then back to calcite. And important thing, no amorphous phase is detected at the end of the process when we collect the crystal. Okay? So let's describe a little bit the crystals now. Of course, when you add these additives, these organic additives, the kinetics is much lower, and you see some increase of pH. And then you see the birth of kind of polycrystals of calcite like this. But when you grow, when you enlarge this part, when you blow up this part, what you see is a kind of layer organization. So you see layers of calcite, which are quite well defined, like this. If you go and see much closer, what we were interested in is the nanostructuration. And nanostructuration, actually, you can see it. If you blow up these layers I just show you, you see that now there are some grains which appear, and you can measure, and the average size is around 16 nanometers. Of course, it could be an artifact if you think of gold sputtering when you increase that. So we also went to AFM, and AFM, as I told you, is interesting because you can distinguish between the properties of the mineral and what could be around the grains. And this was done here, either by topography or by phase images. And by phase images, you definitely recover the same structure of nano domains, which are surrounded by a kind of cortex, which is of different mechanical properties. And that we believe to be organics mostly, because as I told you, we cannot detect any glassy calcium carbonate by infrared. Even more interesting, when now you try to make TEM, you could observe some of the layers by TEM. And if you make electron diffraction, these layers are diffracting a single crystals. So you seem to have recovered the same kind of organization that was present in NAC, which means that you have these nano domains, and they are also oriented in the way that they diffract like a monocrystal. So if you want now to compare with NAC, what are the features we have recovered and what are the features that we miss? Of course, this kind of crystal is not at all resemblance on the whole of the NAC, of the beautiful wall of bricks that I showed you before. And of course, we only have here this soluble part of the organic matrix. And what's interesting is there was also pictures which were taken from sick pinctadas. Pinctadas could get some diseases where this disease was known to damage the insoluble organic part of the organic matrix. And actually, they got this curious shape, which is not so far from this one. If we go at the nano scale, which is the most interesting part we were looking at, we have to compare our picture to pictures of NAC. Well, the scale is a little bit different, but you see the kind of organization we have is also dependent on the age of the object. For juvenile NAC, you get this organization of nanograins, which quite large cortex. For older specimen, you could see that kind of organization. So just to finish, I don't know how much time I have, but I'm
5 minutes is okay.
Okay, so I just organize a short discussion of the result. Of course, we may wonder what is the origin of this nanostructuration. And we have these grains. So do they form as aragonite nanoparticles? I talk about, of course, of the true NAC, not of our system, because it's calcite nanoparticles. Let's say, do we have, from the start, crystalline nanoparticles, or do we have glassy domains which crystallize afterwards? I took this picture from Helmut Kölfen, and of course, this was described in his paper by nucleation clusters, which make elementary bricks. These bricks have the choice, as it was explained by Denise before, either to grow and to make single bigger crystals, or then to assemble in what is called mesoscale assembly, like this. But you can also grow amorphous particles, as we learned before. And these amorphous particles can transform later on as crystals. So the question is, of course, as glassy precursors are largely documented in calcium carbonate growth, and they were also observed in some calcitic prisms, we may wonder if these nanoparticles are not originally glassy nanoparticles, so particles which are mixed between calcium carbonate and organics, which crystallize afterwards, and by crystallization they have to expel organics, which is now go around the crystalline core. And the question, of course, is how the orientation is preserved. In this process, if you think of these glassy domains expelling organics, which now crystallize, they have to keep the organization in order to diffract like a monocrystal. And for this, we lack a precise mechanism, of course. So the conclusion, so this soluble organic matrix seems to be able to induce an oriented crystallization of nanodomains by itself. So it seems a key ingredient for that. Of course, we don't recover aragonite polymorph, and many studies claimed in the past that for that you need insoluble, the insoluble part of the organics, or maybe smaller proteins that we cut in our process when we recover the organic part. The ordered sheet-like organization needs also probably some sequential deposition. Remember that here we lack these kinetic effects of deposition of nacre. When this mineral grows, it goes in a very timely, ordered way, because you start by depositing calcite, then at some point you switch to aragonite, and it goes layer by layer with time. Of course, our process is not at all like that, so we lack this temporal control, definitely. So a candidate model could be that you have some glassy intermediates which then turn into oriented nanocrystals. And for that, we need to go to new directions. So a first thing is to better quantify the distribution of the crystallinity at small scale. So something we are starting to do thanks to new method by synchrotron coherent imaging in order to get really the distribution of the organization at small scale. And kinetic studies are, of course, needed also, not only getting the final crystals, but trying to see, as we saw in the gold experiment, the growth of the calcium carbonate crystals with time in presence of organics. So with this, I will thank my collaborators. This work was mostly done by Eric Tseng during his postdoc in close collaboration with Corinne Chevalard, people from Orsay, and of course we have collaboration at Soleil and Institut Fresnel for all this imagery technique. So I will thank you for your attention.